Hello everyone. Have you ever wondered how a new medicine makes its way from a lab into our hands? It's not a magic. It's the result of a long, careful journey called as clinical trials. Today, let's break down the phases of clinical trials in step by step in a easy way. So, what are clinical trials? Clinical trials are systematic studies. It is conducted on human subjects. Why it is conducted? To evaluate safety, efficacy, dosage and side effects of a new drugs or treatments. So these trials are essential. They are essential for regulatory approval and for a successful drug development. Phases of clinical trials are broadly divided into four main stages, that is phase 1, phase 2, phase 3, phase 4. Before that, there is a very important step known as preclinical phase. It means before clinical trials we are doing this study, means before humans we are testing it on animals or in we are doing it in lab. So what is the purpose? Testing the drug on animals and cell culture, and it is not officially part of the clinical trial phases, but it comes before the clinical trial, so that's why preclinical phase we says. And to assess toxicity, pharmacokinetic, pharmacodynamic in animals and cell cultures. So what will the outcome? If successful, if the results are promising, then an INDA, investigational new drug application, is submitted to regulatory authorities like DCGI or US FDA. Then we have our phase 1, which will be first in human trials. So the humans are involving from here. Human participants are been tested here, started from here. So the objective, till here we have been testing in lab or on animals, but from now we have to test our drug on humans. So our objective is to evaluate safety, tolerability, pharmacokinetics and pharmacodynamics of drug in humans. Subjects, how much sample size we are including? 20 to 100 healthy volunteers we have to include in first phase. Rest all the phases have patients, but in special conditions, in treating the cancer or in conducting the cancer trials, we have to include patients. So our key points are to find the MTD, maximum tolerated dose, to study ADME, absorption, distribution, metabolism and excretion, to identify initial side effects and to establish safe dosage range. So the duration of this study includes several months to 1 year. Okay, so some says that it is for 5 years, but it is corrected in later. uh, so several months to 1 year will be the correct one. Now we have phase 2. Phase 2 में we are doing therapeutic exploratory trials. So objective is to assess efficacy and further evaluate safety in patients. Here our sample size is 100 to 300 patients, not the healthy volunteers, as we have taken in phase 1 healthy volunteers, but here we have to take the patients with the disease condition. Key points are: it is conducted in RCT format, randomized controlled trial, and it is to determine optimal dose and uh, to provide treatment regimen. It is often divided into phase 2A and phase 2B. A, B may divide. We have been divided into 2. While 2A focuses on dosing, on the other hand 2B focuses on efficacy of drug. Its duration is several months to 2 years. Now moving on to the phase 3, it includes therapeutic confirmatory trial. So the objective is to confirm the efficacy and to monitor any adverse effects in large population. So in large population means sample size, in phase 2 we have included 100 to 300, but here large population means 1000 to 3000, only 10 we are increasing, but we can take more patients also. Key points includes to compare new drug with the standard treatment, as we are confirming that therapeutic confirmatory trial, so we have to compare new drug with standard treatment, and to evaluate drug in diverse population across multi-centers. It means conduct multicentric and international trials, and to collect data for new drug application. The duration of this phase 3 will be 1 to 4 years, and after getting all the data we have, if the drug is a good candidate to treat the disease, we can file NDA, new drug application, to the regulatory authorities. Moving on to the phase 4, which is known as PMS, means post marketing surveillance. Now when the drug has been released onto the market, even though it is safe, we have to monitor its uh, side effects, adverse effects, while it is on the market. So the objective is to monitor long-term safety and real-world effectiveness, and ADRs and ADEs we have to monitor. Subjects we can't say, because thousands of patients are using it in real world. Key points is to detect rare and delayed ADRs, to identify drug interactions, and if there are anything which is causing the side effect, then the authorities can change the label or issues the warnings, or the authority has the power to withdraw the drug if it shows risks serious risks are found, so they can withdraw it and terminate that. So it's a quick recap. We are in on my left hand side, it is phase 1, 2, 3 and 4. So what will the sample size, we are including phase 1 20 to 100, then followed by 100 to 300, then we have to increase 1 zeros here, that is 1000 to 3000, then here in phase 4 thousands. Main focus in first phase will be safety and dosage, then second phase we have to find the efficacy and side effects, then we have to confirm that efficacy by comparison, then we have to long term monitoring in phase 4. In phase 1 we are using healthy volunteers, but in rest all the patients, and that's how a drug travels through the clinical trial phases, from a small group of volunteers to thousands of real world patients. So understanding these phases is crucial, whether you are a pharmacy student or a healthcare professional or just someone curious about medicine development. If you found this video helpful, hit a like button and subscribe for more videos like this. Till then, we'll see you in the next video. Study hard.